Welcome to the second EUFORGEN webinar related to the Ash Dieback and its impact on the Ash genetic diversity. This series of webinars will be the basis for the work of the EUFORGEN community to define approaches and for the best management of the network genetic conservation units in case of biotic outbreaks. EUFORGEN, the European Forest Genetic Resource Programme, is a cooperation program on the conservation and sustainable use of forest genetic resources in Europe. EUFORGEN is an implementation mechanism of the Forest Euro process, the high-level forest policy interface that's aimed to develop common strategies on how to protect and sustainably manage European forests. Today we have three guest speakers who have kindly agreed to share their knowledge and research scientific findings on the genetic basis of the sensitivity, the tolerance, and the resistance to Ash Dieback, and also guide us to the recent work on tree breeding and findings related to that. So our first speaker is Richard Bucks from the Royal Botanical Garden of Kew and also the University of London Kew Mary. And he will introduce us to the three genetic bases of the sensibility and tolerance of Ash Dieback. Richard, please, the floor is yours.
Thank you very much for the invitation to speak to EUFORGEN. It's a great privilege to be speaking for you today. And I'm just sharing my slides, I hope. My slides are just loading up, it's telling me.
Yes, we can see it. Thank you.
Great. Thank you very much. So we've been working since 2012 on ash tree genomics. Our website we recently revamped. So if you go to ashgenome.org, you can find information about our work. I'll have to cover the ground very quickly today. We published the ash genome for the first time in 2016. But then of more interest to this particular seminar is a paper we published in 2019, Genomic Basis of European Ash Tree Resistance to the Ash Dieback fungus. And there we're using the word resistance in a very broad sense to mean low susceptibility. We're not taking a view on mechanisms there. This work was based on trials set up by Forest Research. They took ash saplings from nurseries in the UK and planted the trees in sites in the southeast of England where they would be exposed to the Ash Dieback fungus. We then scored the trees with a system devised by our colleagues in Lithuania. And you can see there the different provenances on the horizontal axis of these trees, mainly from the UK, but some from France, Germany, and Ireland. And you can see the different scores. So every provenance did have some trees that remained healthy. This was in 2016 when the trees had been in the ground for 4 years. So this is just after 4 years. Of course, subsequently, more of the trees that then were healthy have become unhealthy. But this gives us a snapshot of which trees were remaining healthy for longer. We then wanted to do a genome-wide association study. And in order to be able to do that with our funding, we had to do pooled sequencing to get allele frequencies in our populations. And that meant that we could only look at the most healthy trees and very diseased trees. We couldn't treat the trait as a continuum. We had to sort of impose a binary restriction. It's not that we think this is a binary trait, but for the power of our GWAS, we had to just take the most healthy trees and unhealthy trees and compare them. And so we did that. The total number of trees involved in the GWAS was 1,250. And this enabled us to identify loci in the genome of ash with significant associations with the health of those trees. And looking at the protein coding genes annotated close to some of those hits yielded potential identifications and functions of those genes which seem to fit with them being involved with health under Ash Dieback. For example, a plant FBOX gene in this case, and a gene involved in salicylic acid signaling in this case. And so we thought this is plausible. You know, we have plausible hits in the genome for Ash Dieback resistance in the broad sense. We then used a genomic prediction approach to see if we could predict the health of a tree based on its genome sequence. And when we looked at a group of trees that hadn't been included in the original GWAS, we had 67% accuracy of assignment to healthy versus unhealthy statuses. And so the genomic prediction was working reasonably well. We couldn't tell for sure how healthy a tree was going to be, of course, because there's lots of environmental effects and many genes involved, but we did have enough accuracy for a breeding program. In a crop breeding program, a 67% accuracy of assignment would be seen as useful. And if we just wanted to take the top 20% of our breeding values in our population of trees, then the likelihood of them having the correct health status was much higher. So if we did a breeding program just taking the very top breeding values, we would have more confidence than if we took the top 50%. Subsequently, we've reanalyzed some of this data, and this is unpublished analyses. So one thing we've done is the Bee Forest Consortium made the 4-tree array, which includes ash SNPs, and we provided the identifications of the 2,500 best SNPs from our studies, which enabled us to give 67% accuracy of prediction. And the Bee Forest Consortium sought to include all of those SNPs in their chip, but they could only include 1,633 because of the practical constraints of the array. Unfortunately, when we then tried to do predictions based on the ones that had actually been included in the array, our predictions were not very good. And we also reanalyzed the Stocks et al. Data, looking at all of the SNPs in the Bee Forest array, and we couldn't get good accuracy of prediction from that either. So we were quite disappointed about that. But I think it's telling us something interesting, and that is when you're making an array like the Bee Forest 4-tree array, you have to avoid areas where you've got repetitive sequence. You have to avoid sequences that are very similar to others. Otherwise, the technology doesn't work. So this might be a hint to us that some of our SNPs are in regions where there's gene duplication and perhaps structural variation. And that's something we've looked at a little bit more. Here's another analysis we've done. Our colleagues in Poland have released a chromosomal level assembly for an ash tree. And so we reran our data on that because it's a much better assembly than our one. And the results are somewhat clearer, but also somewhat different to the results we had before. And some of the differences might be due to structural variation between the Polish reference tree and the British reference tree that we used before. So both of these strands are telling us maybe structural variants are important. Here's a comparison between our original Manhattan plot and the one reanalyzed using the Polish genome. You can see this, it's quite different. And so we've had a preliminary look at the effect of tandem duplications within the genome on Ash Dieback resistance. And we're using our Stocks et al. Data, we have found some potentially significant hits where duplications within the genome are involved in the health of trees. And this is something that we're following up, as I'll tell you in the next few slides. So this is a new project. It's being led by Laura Kelly, who's been involved in Ash Genomics since 2012. And it's funded by DEFRA under part of a new Centre for Forest Protection. And we're doing a major update on our reference assembly. And we're also sequencing with Oxford Nanopore 50 ash trees from across the UK and Europe. And this is the results for the first 7 individuals, much better genomes actually than our original reference genome that we published in 2016. And we want to turn this into a pan-genome. A pan-genome is a reference that incorporates duplications and big deletions and insertions that are found among the genomes. So we want to understand how much structural variation there is in ash genomes across Europe. And we think this will help us in our predictions of Ash Dieback susceptibility. So in conclusion, we've identified SNPs associated with health under Ash Dieback. And genomic predictions based on these SNPs could be helpful in breeding programs. But we think there's increasing evidence that structural variants are also likely to be important. This is much larger scale variants among genomes than just single base changes. And we're now investigating these. So I'd like to thank our funders, mainly the Department for Environment, for Food and Rural Affairs, DEFRA. Also NERC, the Natural Environment Research Council, funded a lot of this. And please do look at our website, ashgenome.org. Thank you very much.
Thank you very much, Richard. We have allocated a time slot at the end for an interaction and a discussion altogether, but I will like to give the possibility for people to ask you specific questions on something in your presentation in case something was not clear or they need some specific detail. And again, the discussion ideally will happen at the end after the other 2 presentations. So anyone to ask questions, you can either type in the chat or ideally raise your hands and you can just very briefly ask. Okay, I see one hand up.
Hi, Richard. Hi, Richard. It's Trevor here. I was just wondering if you have any view on the role of receptors or receptor genes in resistance to Ash Dieback.
We haven't been drilling down into the mechanisms involved. Our aim has been to be able to inform a breeding program. And as long as we have a genetic marker that's associated with health under Ash Dieback, we don't really mind what it's doing. If we know it's there, we can use it for selection. So the level of drilling down we've done has been very limited. We did a little bit initially just to see if our Manhattan plot was making sense, but we haven't followed that up. It would be something interesting to follow up, but we don't really need to know what these loci are doing in order to be able to use them as markers for a breeding program.
Okay, thank you very much. Then I guess I will suggest that we continue the discussion later on. And now we move to the next 2 presentations. So the next topic will be on the Ash genetic resistance research and entry breeding that is ongoing. And we have 2 speakers. The first one from the Thuenen Institute in Germany, Ben Bubner. And that, yes, I can see that you already started to share your screen. Please, Ben, the floor is yours. You're muted, sorry.
Now
it's good. Yes, thank you very much.
Yeah. Hello, everybody. Thank you for the invitation to give us talk and everybody who attends today. Yes, my name is Ben Bubner and I'm from the Thuenen Institute of Forest Genetics in Germany, and I will talk about the German efforts to select tolerant trees, ash trees. And so I have so I will say something about Ash Dieback in Germany, say about the structure of ongoing projects in Germany, talk about selection of plus trees, and give also an insight to the QTL analysis of tolerance. Fraxinus excelsior, there was an important forestry in Germany as a broad-leaved tree 24% of forested area in Germany is covered by Fraxinus excelsior. And as everywhere in Europe, the problem is that we observe a dieback of shoots and whole trees and root collar necrosis. The first observation we had in Germany in 2002, and it moved on to Bavaria and Baden-Württemberg by 2009 and 2010. Today, our forest stands are affected, but we observe single trees that stay healthy, and we hope that they are basis to select tolerant ash trees. There have been some initial research initiatives in the federal states in Germany, and it was decided by the German Federal Ministry of Food and Agriculture and the German Federal Ministry of Environmental Protection to bundle these efforts and to set up a concerted action called Frax for Future, that itself consists of 5 joint projects, and we are altogether 28 projects. So a lot of power, a lot of scientists. And in Frax Connect, they coordinate the project, and we have also an economic part. In Frax Mon, the projects deal with monitoring of ash dieback, including terrestrial monitoring plots, but also remote sensing. In Frax Pat, the projects deal with the pathology of Hymenoscyphus fraxineus, and in Frax Gen we deal with the genetics of ash, and we select the tolerant trees. And Frax Silver works with natural regeneration and how it can be used in silviculture to preserve the ash. And the general aim is to save ash as a forestry. We learned last week that ash will survive as a shrub, but we want to have trees. In Frax Gen, we are 7 partners, and we have also 7 topics, but they are not distributed one by one. The most important topic covered by 5 partners is the plus tree selection. And then we also have in vitro cultures as another way to preserve biodiversity and clusters. We analyze phenolics and biomarkers in cooperation with projects from the monitoring group. And we have a QTL analysis that I will shortly talk about. We also develop new markers based on transcriptomics, and we want to validate the known markers. And one method would be also to try the bee forest slip chip that Richard already talked about. And also population genetics is a topic not only for the plus trees, but also for the trees in the monitoring plots. The plus tree selection goes on in 9 federal states. In Mecklenburg, Western Pomerania, the selection program is already finished. It was from 2016 to 2020. The approach there is also the blueprint for the ongoing selection program on adult trees. And we have already here a clonal seed plantation with 121 genotypes. And the white spots in the western parts of Germany is just because nobody from there applied for the joint project. In the whole concerted action, we use a unified key. It was developed and is also used by the monitoring people and also by the pathology projects. And we have 6 damage scores. And in Frax Gen, we select adult trees that have a damage score of 1 and 2. And the damage score 0 doesn't exist in Germany anymore. At the moment, we have selected 631 plus trees. And here you see also the 5 institutions involved with it. And the first round of assessment was in 2021 in summer with a subsequent winter assessment, and we grafted the trees, the selected trees in 2022. And the second round of assessment is still going on, with grafting planned within the next weeks. We grafted about several thousand grafts, 20 gametes per genotype. And the grafting success per genotype is almost 100%. But not all gametes of a genotype start to grow. And here the success rate is 50 to 90% among the 5 institutions, depending on institution. And what is important in our project, we test every plant, also every graft, we test with 2 test systems, with an agar plug test system with 2 isolates of Hymenus vaccineus, and also with an ascospore test, which means we collect the pathiols from the previous year and put them in summer under the pots with the grafted trees. And we assess in autumn the leaf spots and the shoot necrosis later in autumn and winter. And what is important here, the assessment rate is now done for the grafts of 2022 of last year, and we can confirm the plus tree status for 40 to 90% of the genotypes, also depending on institution. And so 40% is quite low because we still don't know how to handle it. If we still plant them in a seed plantation, and we have to decide it very soon because the planting of the clonal seed plantation will start within the next weeks. We also plan progeny trials for plus trees, but first of all, only half of our trees are female. That means they have fruits, and droughts caused many sterile seeds in last year. We had less seeds than expected, and yes, we sow them as green seeds. And we will determine the germination rate this spring. But we know already by now that for establishing progeny trials, we won't have enough plants, and we will start collecting seeds this year again. And hope it will work better. And here now comes the outlook to the QTL analysis. This is just another method to explore the genetic architecture of Ash Dieback tolerance. And the usual method in tolerance research would be to have a tolerant tree that you cross with a susceptible tree. And then in the offspring, you see the variation of a trait, and you connect it to genomic data, and then you can do a mapping. And because it is very complicated to cross ash in the field, we decided another approach. We selected 5 trees in stands, and these 5 mother trees, healthy mother trees, have been surrounded by decaying trees. And so we think that when the pollination has occurred, it was from a susceptible tree. And all what we had to do to analyze the seed the harvest for full siblings. In the end, we had for each progeny 960 plants, and because this is about 4,300 plants altogether, and this would have been too much just for funding reasons to do a complete genome sequencing at the beginning. So we screened them with a reduced set of microsatellite markers, all the 4,300 plants, and the parentship test of colony. And we had full sibling families with membership numbers between 50 to 160 plants. And altogether, we selected 950 progeny for an Illumina resequencing. Now you see the number of markers is much higher, 1,400,000. We did the principal component analysis and to see if we could confirm the colony results. And in the end, it turned out that we have 4 large families of full siblings, each from another mother tree. For us, the method worked very well. So we had a breeding without crossing approach. And the first step of doing a microsatellite analysis was a very good step to reduce the number of genomes we had to sequence. And now we have good full SIP progenies with genome sequences that we can use as a basis for QTL analysis. Here you see a first result of a full SIP progeny with 161 plants, for which we had also the Ash Dieback data. The SNPs I have used here are 2,500. And you see that we have here signal at chromosome 8. This is in opposition to what we've seen before with Richard Bucks. I have to say about that the phenotyping occurred after the first year in the field trial. So we planted in 2021. So infection was 2021, the possible infection, and we screened last winter. We will repeat the screening, so the screenings every year. So Richard Bucks did screen it after 4 and 7 years. So that is very well possible that the phenotype still has to develop to give results, substantial results in this analysis. Yeah, and now already I'm at the end, and I thank you for your attention.
Thank you very much, Ben. I think we will have just time for one question, and there is also already something in the chat that is asking if you have a number in mind for +3 selection and for how many years you will continue to have selection, including trees in your collection.
It depends on the funding. Also actually we are finished at the moment. So these are the numbers of +3s that I showed you. Won't change much more anymore if you don't get more funding.
Okay.
Yeah.
Thank you very much. Then we can move to Heino Konrad from BFW in Vienna, Austria, that he will share also some of the recent findings in the research, including tree breeding. I think, Ben, you need to stop sharing.
I did, isn't it?
Yeah, now it's working. And then Heino, the floor is yours.
Thank you very much. Can you see my, wait, I have to put to full screen.
Yes, so, now it's in full screen.
Can you can hear me? Great.
Yes.
Thank you very much for having the opportunity to present our project here. So we call it Ash in Distress. It's a breeding and conservation effort for ash in Austria. It's a cooperation project between my institute, Austrian Research Center for Forests, and the University of BOKU Vienna. And of course, this is not my own effort. So I have to say a lot of the work presented here has been done by Gregor Unger, of course, Jan Aglaya Schukala, Thomas Kirisic, Silvio Schüler, and the former leader of the project was Thomas Geberer. So I will just show you a few slides about the symptoms. I think we all are aware how the disease looks like. So you have the dieback in the crown, you have the dieback of the whole tree, and in association with this root collar lesions, eventually, and in association with the root killing fungi, the root is damaged and the trees falls down, can topple off. And in this way, the disease has also created a lot of common interest, not only in the forests, but also in the common public. So the disease was first detected in Austria in 2005. In 2007, Thomas Kirisic was able to isolate it for the first time. At that point, we were still hoping that it will stay a local problem in some areas, but by 2012 it had spread to all over Austria, so it was basically present in all the occurrences of ash in Austria. and of course was causing a lot of damage. But there was hope, because in many stands you can see still some ash trees which look relatively good in comparison to the rest of the population. So there was early hope for a genetic component to the resistance. And then Thomas Kirisitz, very early, already in 2011, he started to evaluate some of the clones in one of our ash seed orchards. And he could show that over the years, this disease incidence pattern stayed constant. So the same clones stayed healthy and the same clones deteriorated in the disease occurrence. Maybe you note down here these two clones, the number 43 and the number 36, because Thomas then did a smaller experiment with offspring of these clones, and he could show that that the healthy clones also, the healthy clone also had the healthy offspring and vice versa. So there was a relatively high heritability to the resistance trait. And this eventually has then led in 2015 to the to our project Ash in Distress. So it has run from 2015 to 2019 as in the phase 1, and currently we are in phase 2 of the project. So it's a long-term effort, and we have to be very thankful to our donors to keep the funding persisting. So what have we been doing? We have been searching for those resistant trees all over Austria, starting in 2015. So on the map, you can see Austria, and all the green dots were the populations which were successfully harvested for resistant mother trees. So in total, out of the more than 800 places which were we were notified about, 435 sites were actually visited, and from there we were able to collect seeds from 715 mother trees. So this is a quite big sample covering most of Austria. How did we select the mother trees? Of course, we were looking for trees with a healthy crown, with no or very little disease incidence in comparison to the rest of the stand. And of course, we were also not looking for, we were only looking for trees in the forest. So field trees outside of the forest were avoided, of course, because there's just not enough inoculum around to get them infected. And we were also selecting younger trees. So the maximum DBH was around 30 centimeters, because it can be difficult to assess the disease incidence in older trees. So yes, and in this way, we eventually were able to raise about 35,000 seedlings out of this seed material collected from the more than 700 mother trees. And these were then raised in our common garden in Thuenen. And so the whole plot was organized in 4 subplots. This was due to the fact that we did the seed collections in 2 seasons. The first one was in 2015, and the second in 2017. And because the ash seeds, they germinate over a longer period, so we had to organize for each collection year, we had to organize the common garden experiment in 2 plots. So we have these resistant plots, R1 to R4 in total. And the total, the plots are, the plants in the plots are totally fully randomized, planted. So this was also a huge logistic effort. And this was very well organized by Gregor Unger and the nursery people. So That was a big thing. Then we observed many traits. Most important, of course, the height development of the seedlings. The flushing behavior was assessed. And then, of course, the Ash Dieback incidence, which was assessed in summer after the first year of growing in the plots. And this was done for all the 35,000 trees for three times for each tree. How did we score the disease incidence? This was also done following a scheme developed by Thomas Kirisitz. So we had the disease class 1, which is basically healthy, no dieback. And disease class, the damage class 6 was 100%. So that means the tree was actually dead at the observation. And this was the percentage shows the damage of the woody parts. So the leaf lesions were not scored. So how did this look like? So as said, we have already selected from field-tolerant, field-resistant trees. So in the end, we had a percentage of about 20% healthy offspring on average from all the plots. This goes for R2 and R1. In the younger plots, in last year we had less disease incidence, so actually it was around 30%, but we expect that it will be eventually around 20% as well, because last year was a very dry summer, and this was maybe helping the ash not to get infected by the fungus. So yes, what are our next steps to bring the ash back to the forest? So we are still in the phase of selecting and propagating the best clones. The next step will then be the provision of the clonal material to the forest managers and to install the first plantings as field trials with clonal material to assess the selected clones further. And in the step 3, there is the establishment of the seed orchard with the elite clones. And as you have seen, we have quite a lot of lots of resistant clones. So the plan is to have more than one seed orchard as a result, also taking into account the very diverse geography and also the elevational layers of Austria. So at the moment, we are still in the selection and propagation of the clones. So we are further observing the disease incidence. We have roped out all the susceptible clones. So that means only the healthy trees are remaining. But these we also have cut back to avoid competition between the different clones, but also to facilitate the vegetative propagation of these clones. So we have quite a big set of clones. So we, besides the disease incidence, we also have tried to include growth performance in the selection as the selection criterion. And of course, we are also avoiding to select too many related clones. So we also try to conserve the genetic diversity of the natural populations. So yes, at the moment we are propagating these clones by soft cuttings in summer and by grafting in the coming weeks. So far, we have collected, or we have grafted about 200 clones, and these are ready now for further analysis. So as similar to the approach that Ben showed, we will also do artificial infections with Hymenocyphus, but also artificial infections with Armillaria species, because these Armillaria species are very important when it comes to the root the root disease and then eventually the falling down of the trees. We have also in cooperation with Richard's group, we have been also analyzing the genomics basis of the resistance. So we have run this axiom, this 4-tree SNP array, which covers around 13,400 SNP markers from the genome. And what we have done is similar to the approach that was initially also done by Richard's group. And we have analyzed around 600 pairs of SIPs. So we had siblings from the same mother, but with high and low disease tolerance. And these were the basis for our analysis. So the first result was that we have quite a strong population structure actually in Austria. And this is also something that Richard has found. There's quite a strong structure in the ash population in Europe. So we have a cluster in the west of Austria and a bigger cluster that covers most of the rest of the country. But we also have a cluster coming in the east, this greenish blue cluster, which is probably introgression from narrow-leaved ash, which is touching Austria in the right-hand corner. Then we have also analyzed the SNP data towards the resistance genes, let's say. So we were able to define the groups based on the SNP data, the susceptible individuals and the resistant individuals. And we have also already discovered a couple of SNPs, candidate SNPs, which could be effective to show the resistance, but here the analysis is still ongoing. And this part of the analysis is done by my colleague Aglaya Shukala. So not to forget, as said, we have also this narrow-leaved ash, a little bit in Austria, but it's an important species in Eastern and Western Europe. So we had another smaller project in resistance breeding also in that species. So we have selected around 100 clones, which we now have in a clonal archive, and we have collected seeds from those also. So we also have installed field trials from that species, and that project ended. But of course, we will continue to monitor the plots and continue also the breeding effort in the narrow-leaved ash. This was funding from the Rural Development Funds from the EU. Yeah, and with this, I come to the funding and to the saying thanks. So we had generous support from the Federal Ministry of Agriculture of Austria, from the Provincial Forest Services, from the Chamber of Agriculture. And of course, thanks go out also to our whole team, especially, of course, to Gregor Unger, who was is still the heart and the soul of the establishment of these field plots and the whole setup. And of course, to the people in the nursery who have done tremendous work installing all the plots and measuring the traits. So thanks to them, and also thank you for listening. And I hope there are some good, nice questions.
Thank you very much, Heino. Indeed, there are many questions popping up in our chat. Before I go into the more general part, I would ask if there are any specific question for Heino now. Otherwise, we can start addressing some of the questions that we have in the chat already. I suggest that you you can just raise your hand and then I will moderate the discussion. When I will give you the floor, kindly introduce yourself with name and affiliation so that we can better figure out the interaction that we have. So again, any specific question for Heino? Then I suggest that I could either start going through the various questions that we have in the chat, or if you the participants online, you can just raise your hand and I will give you the floor. I will have myself a question. Oh no, I see that Jerry Douglas. Jerry, long time don't see. Happy to have you online. Please. I don't know. I cannot see. We cannot hear you. In case you, but you can start speak.
Oh yes.
Okay, now we can.
Thank you. My suggestion is for Dr. Bubner. Can he suggest the best time, the optimal time of the year for selecting healthy trees?
Can you hear me? Yes.
Yes.
Also actually, we do it in a two-step approach. We do it in summer, and then we reassess the trees in winter time. Yeah, because in summer we look just for how healthy the plant looks like, and in the winter time we can also look at the structure of the twigs. Yeah, because they also tell us something about the health status. So we would we do both times, 2 times in the year. And we combine it, the data from the winter and from the summer. Okay, thank you.
Thank you very much. And then, this is a little bit of a challenge. Matthäus Linzebritz. Sorry, I will need to ask you to kindly repeat your name because I'm sure I've killed it.
Hello, Matthäus Linzebritz. Yeah, but it's fine. It's fine. Thank you for the presentation. I have more of thoughts than the questions to maybe all of you. I mean, the selection of the plus trees just based on the observation in a single year. And do you have a plan to check it, like the plus tree selected in nature over the years? Because it can be just this single time it's kind of resistant, but it might change. At least I observed that it has been changing a lot. And then I have a question to Konrad, like this planting pattern and planting spacing, it was very tight. I mean, it was very dense. Are there any limitation of that? Is the transmission of pathogen higher or lower than because it must have an effect.
Actually, yes. I mean the spacing was, if I'm not mistaken, 80 by 20. So it was very close. That's right. In the beginning, that was actually good, I think, because then we had like a closed canopy of ash quite quickly. And in this way, we had lots of inoculum there. So in the beginning, we put some, let's say artificial inoculum there. But during the trial, it became a lot of natural inoculum there. But you're right. In the end, after 3 years, we now, as I said, we had to cut back all the trees, or even the good ones. Otherwise, there would have been too much competition between the different clones. So yes, and the spacing initially, of course, it was a lack of space in general. That's why we had to go for rather close spacing, the common garden.
Thank you very much, Heino. And then we have Tanya online with another question. Please, Tanya.
I was wondering, this is, I guess, like a general question. How much is known about the genetic variation within the pathogen? And when you are selecting for resistance, is that general or against certain strain of the pathogen? Or is this like a universal distribution that is the same strains of the pathogen all around?
much to answer. I think last week, Michele clearly had a nice talk about the genetic variation of the pathogen. I think in Eastern Asia there's a higher variation, but as you know, the sexual reproductive pathogen, so there's lots of diversity created all the time. So, so we worked with natural infections. So at least let's say we had a realistic situation. Maybe Thomas Kirisitz or somebody is here who can, or Ben, I don't know.
Thanks.
Yeah, maybe can I answer to it?
Yes, please go ahead.
So I think the problem is really that we actually have no strains because the fungus is propagating by spores. So we have a lot of variation either by myospores, from sexual reproduction. So actually each fruiting body produces thousands of individuals. Yeah, so that is, you have actually no strains, and this is really a problem. I've seen some questions regarding our inoculations. Of course, the inoculations are a big problem because we select only one or two genotypes, but the natural infection are hundreds, thousands of genotypes every time. And so I think actually the more promising approach is always to use the natural infection, or at least something that comes from ascospores, as Heino said. I think this is a better approach in the moment, at least. Yeah.
I mean, we will also do these artificial infections. For that, we have, of course, strains, but those are also aggressive strains from the field, which have been kept virulent, let's say.
Thank you very much. And I think we have time for maybe one more question. Meanwhile, I can start reminding you that the previous presentation is already available through the EUFORGEN website, and this webinar as well is going to be made available ideally today, later this afternoon, on EUFORGEN YouTube channel, and then again just going to the homepage of the program, you will have a direct access to the recording. I see that there are really many questions in the chat already happening. What the speakers already indicated they will be available later on in case you have something, some specific question, also to have some interaction. I can see there is a lot of interest also in some technicalities of both the research and the sampling and the management of the plantations. Then I would also like to announce that we have made a small change in the next webinar, because in addition to the plan presentation, natural selection in Ash population related to the spreading of the Ash dieback, Richard Bucks agreed to give an additional speech on the genomic evidence of natural selection by Ash dieback in unmanaged woodland. And this is going to be part of the of next week webinar. I will have one small question myself related to the the idea of all this effort is to bring ash back in the landscape, but the disease itself has caused a dramatic loss in genetic diversity. Do you think that the new population that we will be able to bring in the landscape will be more on a sort of plantation that will need to be closely managed by humans, or you think that we can see again ash coming back as a natural species occurring in the landscape?
Shall I try?
Yes, please.
Yes. In our project, we have tried really to have a broad genetic basis. So we have the clones from all over Austria, which is a relatively small area. And then also in our seed orchards to be developed, we try to have as many clones as possible, still with high breeding value. But what also our silviculturists are doing is that we have to see ash maybe a little bit differently, not as a dominating species, because sometimes it really has become dominating. Those are the stands which are most affected, but you have to mix it with other species and to have little lower incidence of the spores. Then you have also a higher chance to grow them into bigger trees, I guess. So there will be less ash probably in the future. Yeah.
Okay, thank you very much. I don't know if Richard and Ben, you want to have some concluding remarks. Otherwise, then
I think I would just.
Then I take this opportunity for thanking the three of you for sharing your knowledge and your research findings, and for everyone online to be with us today. And see you next week for the next webinar of the series. Thank you very much and goodbye.
Thank you.